Okay, it is my great pleasure to introduce Jennifer Fouquier. I said that right this time?
Fouquier or Fouquier,
Yeah.
either way.
All right, Fouquier. How could I did the first time I met you, I got it exactly right. And ever since then, I've messed it up. So I don't know French. Um, anyway, it's my great pleasure to introduce Jennifer, who was my BMI master's student long time ago. And now has a full-fledged PhD with her own company, which is really exciting. Jennifer came out of industry, had several years in industry. I think it was at least 5 um, before you came to work at San Diego State. And we said, and now I realize, and I remember now vaguely that you didn't have any real bioinformatics training, and you just said you didn't even own a laptop. So for all of you there who are biology students who aren't sure of your abilities, you're in my class and you're learning, it is totally possible to become an amazing programmer, an amazing bioinformatician, coming from basically not knowing any of that stuff. But we knew in our program that someone so dedicated, had so much experience in industry and good letters and all these things would be a great fit for the program. And we couldn't have been more right than with Jennifer. In fact, during your time here, you published, I believe it was 4 papers out of your master's, two first author. And that's just stunning. I mean, that's more than some PhD students. I wish I could have just handed you a PhD after that, because you were definitely PhD material. And as I recall, you took an extra term because you wanted to be even better at the programming and even be a better computational biologist. So anyway, after that, you worked, Jennifer, worked for a while over at Scripps and over at, I think you went to Colorado and worked there for a bit, and then joined a PhD program at the University of Colorado, Denver. Is that right?
Anschutz Medical Campus.
Anschutz Medical Center, that's right. Anschutz Medical Center, which used to be the Cell Sciences Center. And my wife actually got her degree at the old institution before it moved over there. And anyway, had to bear through a PhD through COVID, which is no mean feat. It's a real big struggle, but definitely managed, and did a wonderful thing, which you're going to basically talk about your thesis work and how it actually started your company. And at the end, if you want to, or in the beginning, if you want to talk more about your journey, I'm sure other students would love to help. But I really want to hear about this awesome software and
Yeah,
awesome stuff that you're doing.
thank you so much for the introduction. So I actually started with, I'm talking mostly about my work that was done at Anschutz Medical Campus. And I wanted to start with my acknowledgments. My advisor I've been working with, Kathy, Scott got the better years of me. She actually got some difficult years of my life. So, but I'm really appreciative for the opportunity there. I was in the computational bioscience grad program. Currently, I'm not working. I mean, I am working I started a company, and I'll go over that, but it's very new. I just graduated in May. I was on the bioinformatics training program, a training grant at Anschutz, and I worked with the tech transfer office there for the software. And this was what Scott was mentioning, my career. I have about 10 years of wet lab experience. Much of it was part-time. This is a picture. Look at that old computer right here. That is an old computer. So I started when I was 16. What was that, Scott? Oh, it's muted. You're muted.
I was saying I can't see the old computer.
Right here?
Oh, I don't know. Can you see it, right?
Do you guys see the slides? Are the slides moving?
We can see the
Oh,
slide.
okay. Right
Scott,
here.
Okay, I'm the only one.
Scott looks at that like a normal computer. This is an old computer. I'm teasing. So yeah, I started in the wet lab. And then I went to do my master's in bioinformatics and medical informatics with Scott at San Diego State University. And then I went on to work with Andrew Su at the Scripps Research Institute. And what I wanted to point out here was that I started with microbiome research. And then when I went to Andrew Su's lab, I worked on web development and citizen science projects. So it was really that connection was really helpful for me with this tool. And I just want to thank both of them. And I suppose now I have a conflict of interest because I filed what's called a invention disclosure at University of Colorado Anschutz Medical Campus. And so I wanted to mention that because if anyone has a tool that they think could be commercialized, then you should talk to a tech transfer office, talk to experts, and don't make your code completely licensed for everyone to use, because they can help you there. But if you want to commercialize it and have more people use it, you could consider going this route. And I actually had to pay to use my own software, just so everyone knows. I had to make my own license. That's how it works. And so I'm going to give a quick background. I'm going to go through the beginning part of my research pretty quickly because I want to get into some of the results, which are a little bit more interesting. But I kept a lot of the text here for everyone in case anyone wants the slides. So I worked on the microbiome, and as a lot of us know now, the microbiome is associated with disease. It's the community of bacteria, viruses, fungi, and genes. And we use 16S rRNA gene sequencing to look at the community. It's associated with all kinds of diseases: HIV, autism spectrum disorder, obesity, and diversity is usually related to health. And there is, fortunately, hope for disease treatment through diet, probiotics, fecal transplant. And we're coming across more longitudinal microbiome studies, longitudinal studies in general, and those help to assess changes in the microbiome over time, and they better account for inter-individual differences, which we know are pretty big. And longitudinal studies contain repeated measures or non-independent data. And when we're trying to find a relationship between the response variable and the input variables, when we're looking at longitudinal or repeated measure data, we need mixed effects models. And that's the fixed effects there. The word mixed comes from the fixed effects, which are the effects of interest. And the random effects are the grouping structures not of direct interest, like your subjects. And so on the right, we can see this looks like a negative relationship, but it's actually a lot of positive relationships. And so we need statistics that can help to account for this, the individual differences. And longitudinal analyses can be performed in different ways by comparing one value to different reference points. And it becomes quite complicated when you think about this. For an interventional study, you would have an interesting baseline measurement that you might be trying to change, such as a poor health outcome at baseline. But then in an observational study, if we're looking for fluctuations over time, we might not necessarily have a baseline value. What's a baseline? So that's what started this journey. Was I was trying to identify important features in an observational longitudinal microbiome study in autism. And we had 7 children with autism ages 2 to 11. We had 2 to 4 samples over 13 to 14 months. We were looking at all kinds of variables: gastrointestinal distress, nutritional, 16S, microbiome. And we had a bunch of response variables: hyperactivity, inappropriate speech, irritability, lethargy, social withdrawals, stereotypy, or inappropriate movements. So I had all this data, and I was thinking about this problem with looking at change over time. And like I said, I'm going to go over this pretty quickly, but we had distance matrices, and that's when you look at the entire community of microbes, and you can see how the community changes. And so you can look at the pairwise deltas between samples from each person. And very briefly, when I was looking at the pairwise changes using all pairwise comparisons, not a baseline, we found that there was a bigger change in their withdrawal and lethargy related to a bigger change in the microbiome. But then when I used the original data without making any delta or differences calculations, I found that there was a decrease in inappropriate speech as the number of microbes increased, and that relates to what we see in health. So the inappropriate speech went down as the organisms increased. And so that was using an observational study that got published, but it really made me think about longitudinal data and how complicated it is. And some of the conclusions were that we lost power. I lost power when I was using linear mixed effects models, and the analytics were really cumbersome. I was looking at changes. I was building all these different models with regular linear mixed effects models. I wasn't sure if I was able to capture nonlinear relationships. There are ways you can kind of do that with linear mixed effects models, but I'm not too familiar with that. And I just wasn't quite sure if the LMEs were the best way to do that. There's other complicated methods that would be better, like machine learning methods. And I knew that there was more data coming, and I had an interventional study coming. We had a diet intervention in HIV positive individuals, and we had their outcome data at baseline, and we knew that we were trying to improve their inflammation or their metabolic health from a poor baseline measure. So that was interventional. So then I didn't want to necessarily think about pairwise changes. I wanted to think about changes compared to a baseline value. So looking at change starts to get really complicated. And so figure on the right shows us how feature changes can be uncovered, and they're important in different relationships or in different situations. So when you're trying to, so the top, we have an example of a microbe, a numerical variable. And then here we have a categorical variable. This is just an example. This would be color, blue, green. And then here is the original data. This is one microbe, blue, 3 microbes, green color, 7 blue. As you can see, that's the original raw data. But then if I wanted to find the differences, we could use the baseline differences. So this would be the change compared to baseline right here. And then this is compared to the previous value right here. And then all pairwise comparisons. So then you're taking all these pairwise, and that's what I did in the autism study. And that was what, when I started to look at this, I started to think that I could include categorical data, because I was typically excluding that or using categorical data as grouping variables. So I was able to include both numerical or categorical data in the Delta data sets. And this is really important because things like drug interactions would highlight the importance of categorical deltas. So to treat heart arrhythmias, there are 2 drugs. There are a lot of drugs, but 2 of the drugs you can use are amiodarone and quinidine. And unfortunately, there is a major drug interaction between these 2. And amiodarone has a really long half-life, elimination half-life from the body, 13 to 142 days, I was finding. And quinidine is eliminated in 6 to 8 hours. So they have very different eliminations. So if you take them together, there's an interaction. So that would imply that amiodarone, which has the longer half-life, would have more likely cause an interaction if it was taken first, not if quinidine was taken first, because it has longer half-life. So categorical changes could be important to look at. So overall, feature selection and exploratory analysis in longitudinal microbiome studies have a lot of challenges. There's many variables and changes. I wanted to identify important variables or patterns. The specific question that I was interested in is what features relate to a response or outcome variable. And this can be done in a data-driven manner with less emphasis on prior knowledge, because as I was working with the autism study and the HIV, I realized it's just so hard to be an expert on all these topics. And I didn't feel like I was giving these projects my full attention by doing literature searches and trying to build statistical models that way. So I wanted to do a data-driven project. And I know that there are methods out there. There's all kinds of machine learning methods you can use, but random forest is a pretty easily interpretable method. It's based on simple decision trees. They can be used for classification, for categorical outcomes, or regression for continuous outcomes. And this is an example on the right of how we could classify a subject. If they were 60 years old, go over here, smoker there, other health condition, yes, it looks like this person would be classified as high risk. So you can just kind of see how a simple decision tree, when used in an ensemble of a bunch of different decision trees, because that's what random forests are, it's using a bunch of different decision trees and looking for complex patterns in your data. And it can be used for linear relationships, complex interactions. It can be used for feature selection, not just for prediction. And then they also have a mixed effects software available. So mixed effects random forest, not just random forest. And it can be used for non-normal data distribution and collinearity, or when your variables are related within your data. So overall, there's a lot of challenges. We have dimensionality reduction, and if we want to play around with our data before building models, we need to merge different data sets. We get data from different sources. We get clinical data from people. We have the microbiome data. We have surveys. We have all kinds of data. We often have them in different data sets. We have these deltas over time. It's confusing. We need mixed effects models. So overall, this was just really challenging for me. I was getting very overwhelmed with all the data and that I had, and it was just becoming cumbersome to explain the results. And I looked and looked for tools, but they just weren't suited for exploratory purposes. Some were more for hypothesis testing, but I wanted to perform exploratory analysis and put all my data in. Some look for consistent temporal trends, but feature fluctuations might be interesting or informative. And they often require many time points, but longitudinal studies often only have a few time points. And then categorical features are often excluded. So I was having a hard time finding a tool that would work. So my goal was to develop a feature selection tool and test it on simulated and published data. So this is a workflow diagram of Explana, the feature selection tool that I wrote. It's a workflow. You start on the left with all your different data sets that you have, and you can perform pre-processing on each individual data set separately as you need, using filters or dimensionality reduction methods like principal component analysis, where you can group certain variables together. And then you can merge the data into your original. Can you guys see the mouse? Okay. You can see so you can get your merged original data set right here. And then here I'm creating the delta data sets where you have the first, like I mentioned, previous and pairwise. So you have multiple data sets from your original data right here. And then after that, you can, and here you can add distance matrices, which are popular and like I mentioned in microbiome research, common in microbiome research. And then there's the feature selection algorithm, and that is a step of mostly, I'll go into that, but random forest or mixed effects random forest and other methods. And then it builds an interactive report to compile everything together so that you know, so you have all your data, your figures, and your explanations in one report. So it's all right there. So people aren't clicking around too many places. They still have to click around and look at things, but it's all contained in one report. So it just makes collaboration and communication a little bit more simple. And my hypothesis with this was that I could identify unique features important in different contexts of change, including novel order-dependent categorical features. So the feature selection algorithm that I came up with was to use machine learning methods for non-independent data, repeated measures, methods to find significant features, and methods to rank features and assess their impact on the response. So when we have a group of features, we have 5 features right here, we can use random forest to rank all the features, but and this is what a lot of tools would do. They would kind of stop there and they just say, okay, this is the importance and these are the ranks, but at which point do you stop talking about them? Would you really want to stop? Would you want to keep discussing the least important variable? So we don't know at which point they become insignificant. So then what I also include in the algorithm is a method called Baruda, and that's combined with SHAP, Baruda SHAP. I'll explain those in a second. But what they do is they find significant features. So here we have a smaller set, significant ones, and then SHAP will help to see how those features impact the response or the outcome so that you can understand how the variables relate to the response and improve hypothesis generation. So this is an example of a SHAP summary B-swarm plot. This is what I started with and why I really wanted to get this whole report and workflow going. Or one of the things I started with after random forest was the SHAP. summary plot. So what you have here is this is predicting mortality for individuals admitted to the hospital when they had COVID. So what they have is their baseline measurements. They get all these different measurements from them, and then they can use these SHAP plots to help predict whether or not they're less likely to die or more likely to die. And I unfortunately actually read this plot when my dad was in the hospital because I had all of these values for him. And so I was really having a very, very hard time at this point in my life because I was looking at this plot, looking at all of his levels. And what it was telling me, here you can see, this is an example, high age levels, higher mortality prediction. Blood urea nitrogen, high levels, high mortality prediction. One of the things we all remember, oxygen, if your oxygen is low, if it's super low, you had a higher mortality prediction. So that's how these plots work. Each point represents one individual, and it's that individual's value for this feature. And then it's using SHAP, it takes a combination of all of the data, and it helps show how that variable combined with all the other data helps to push the prediction. So that was very hard because I kind of knew my dad wasn't looking very good, and I sort of regret looking at the plot, but it's interesting, and that's how helpful these plots can be. That's kind of a good example of that. And then Baruda SHAP, what Baruda SHAP does, or Baruda, is when it's combined with SHAP and random forests, it takes all of your features, shuffles them, and then sees how well the feature you're looking at does compared to all the shuffled random versions of the features, so that it uses the same distributions of your data. And then it says, is my feature always doing better than the shuffled version? So it should do better than shuffled versions of all the data. So it does this multiple times, many trials, and then you can see which features are actually significant and which ones aren't. And this is just showing to show, since like my preprint is out, it's in review. It's called a user-friendly workflow for feature selection and exploratory analysis in longitudinal microbiome studies. But this is why I'm saying that it's user-friendly, because the user only needs to use one configuration file to run the workflow. I still think you need to have solid bioinformatics experience or just software experience. So I'm not saying that anyone could use it, but it's a lot easier than a lot of the machine learning methods out there, in my opinion. You can set up your analysis. You say which your response or outcome variable is, if you're what your random effect is, individuals, the sample ID, and then you can go through and mess around with some of your parameters. You have your data sets that you're trying to merge. You can use, Scott was saying that he loves Python, and I do love Python, but I also really like R. I think they're both very helpful in a lot of ways. And so the workflow uses both R and Python. I'll explain that a little bit more. But you can just write scripts in the config file about each file that you have. So if you have your metadata file, you have your microbiome file, you might have your clinical file, each one you can handle differently inside this config file. So you can stay organized with your whole analysis here, where you can include distance matrices. But then this is controlling all those inputs, run the workflow. And there's a lot going on in the backend, of course. And then this is what you generate. You get an interactive HTML report with feature selection results. And it includes, if you have longitudinal data, it gives you the original data and then the results for the first, previous, and pairwise delta data sets. And it's all in one interactive report. There's just kind of some screenshots. So I did test, I'm going to go over this pretty quickly, but I tested the workflow on simulated data. I used a happiness data set. This is just all completely made up using different patterns and trends that would relate to a change in happiness over time. I had 5 time points. 100 individuals, different variables that are really clear, easy to understand. And this is in the paper, just so people can kind of see the results and say, okay, that makes sense. I had health, their health, treatment, their salary, bonus, sunshine, relaxation, just really simple for because machine learning is pretty complex. So I wanted to make it very simple. And the random effect would be subject. So repeated measures on subject. And so you would get the original, first, previous, and pairwise analyses. When I did different versions of this simulated data set, I have the features. That's what the one that I just explained would be just your basic features. Then I added a microbiome data set. I used a package to make a simulated microbiome data set that changed over time with the response variable. And it had, oops, I forgot to include that. It had microbes that were related and not related to the response variable. So it included compositional data. And then I had where I combined these two, the microbiome data and the features, and then I added an increasing number of completely random variables that did not have any relationship to the outcome, because I wanted to make sure that it wasn't just going to find everything as important. So these were I used, I think, 8 or 9 different data distributions so that just to cover all my grounds, I just included those all in there. So it does pretty well. And so these are this is the, I think that's the area under the curve. So that's just how well the tool does at finding the predictive features and not finding the features that were not predictive. And this is just kind of a brief overview of, in the report, I have a feature occurrence figure, and that shows you which features were selected in each of the models. So original, there were 13, it's just a snippet, but first, previous, and pairwise, these are the models, and then these are the features. So here, this is that categorical feature change. If I had something like green to blue, as I showed in that demo, original might think that green and blue are important, which they might be, and they kind of have a low effect. This is the rank on the left, and on the right is the impact. But if I use the delta data sets, I can start to see that this green-blue came out as the 4th most important variable. It's not possible to find green to blue in the original data because we didn't do the deltas there. And then the green, it had a big negative impact on the response. And so I was trying to model that after that drug interaction to just show that if green before blue occurred, there was going to always be a negative impact because I made the data set have that relationship. And this is kind of a little simple example. Here we have green, it has a small positive impact on the response. So when you have the categorical variables, yes would be the occurrence of it, no would be blue or not occurring. And here, this green to blue, yes, it occurred. It has a big negative interaction on the response. So I can see that with the simulated studies. So I showed linear relationships, different patterns in the data. I showed hockey stick patterns, different trends, and then this categorical feature, order-dependent categorical feature that has a relationship to the response. And so, yeah, deltas are needed to find some relationship. So if you don't use the delta data sets, you might miss some important things. They are complex in different ways, and you're starting to look at a lot of data, but you might miss things by not doing the delta data sets. And this is another kind of view. This is that green to blue. In the report, I'm working on more post hoc visualizations. And here you can see that green to blue only occurred at time 3 to 4, where I had that happening. And it was had a negative impact on, it was related to lower values of happiness. So every time that happened, and then this is just more figures. And what I also did was I used and when I made the simulated microbiome data here, like I said, I had 175 were not differentially abundant or related to the outcome variable. Only 25 were, and that's simulated data that I made using a package that makes compositional data over time. It makes a simulated data set. And when I used my Explana compared to Chime2 feature volatility, longitudinal feature volatility tool, they have a feature selection tool. And it's really good, but they just didn't use mixed effects random forests. They used regular random forests and other tools. I think with theirs, you can use different methods, but there weren't methods to do to account for change over time. And then so when I ran my simulated data set through feature volatility, it had a lot of false positives, and Explana only had a few false positives, and it got all of the true positives. So this is just showing, you know, that having the mixed effects models is very helpful when you're using longitudinal data. And so I wanted to show you guys examples. These are fun. These are using real data sets now. So I was looking at bacteria. This is the demo data that they used in Q2 longitudinal. They looked at 40, but it's from a different study. So they had 43 babies. They were sampled monthly for 2 years, and they were trying to find bacterial genera related to month of life over time. So I threw the data into Explana, and what I saw was, here's the results from, this is cropped just so I could fit it on the page, but Q2 feature volatility had selected 61 out of 455 genera. And Explana selected, here's Explana original, first, previous, and pairwise, because this comes from just one report. So this right here comes from one report. These are the results from feature volatility. And you can see here how I showed before, Explana is selecting less with the original data. So this would be kind of your more direct comparison here to here. And then it also shows different rankings of the variables. And then with the Explana right here, this one was pretty interesting. This Blautia was ranked 30th by feature volatility, but it was ranked 6th using the original data with Explana, and then it also came out in the Delta data set. So it looks like this one might be pretty interesting to look at, and that might have been missed with feature volatility. And here, if we look at the SHAP plots, we can see that with higher Blautia counts, and I did add, I think I didn't mention this, but you can use different transformation methods in the workflow, like CLR. As Scott mentioned that, I was kind of going to throw it away. There was some debate in my lab about CLR and its validity because of some other things they had run into. And as you know, with science, there's always, this works in this case, this works in that case. And so I was a little torn on even including CLR as an option in the pre-processing step. But thanks to Scott, I tested it on multiple things, and I was going to miss a lot of things if I didn't do CLR. So for this demo, I didn't do it, but I have been using CLR transformations mostly. And for the simulated studies too, it did look a lot better. There were times I saw some weird results with CLR, but that was something I wanted to go explore later. So we could use Explana to kind of tease that out later. It would be fun. We can maybe do that test, Scott. That would be a fun project for us to work on. But so here you can see Blautia higher abundance is related to later months in time, and the less Blautia is related to earlier months in time. So earlier months. So it might actually be an important organism to look at. That's part of the hypothesis generating feature of Explana or utility. And I looked at the literature, and it looks like there might be, probiotic properties. Here was a mentioning of that 2022 early life events, Blautia. So here we see that might actually be, looks like it might be something to look into. And then I'm going to go over more data since I'm going to be branching out of the microbiome world. I just wanted to mention that I'm not an expert on these data sets. So if anyone hears me say anything, I'm mostly just presenting the data, but it is tempting to kind of make guesses because it's really interesting. But I'm now showing how broad this tool can be. And I don't know all these topics. So I'm just getting data and kind of presenting the findings, but I don't know what they necessarily for sure mean. There could be weird things in the data that I don't understand that or why these patterns are coming up. Um, so I just want to point that out. And it gets messy. You'll see this presentation gets messier and messier just because I'm working with so many different data sets. And so kind of teasing out how to present it. But one data set I wanted to look at was the National Social Life, Health and Aging Project. And it was done on a national scale. I looked at this one because it does have longitudinal data. I'm only going to show the cross-sectional, but that's why I started looking there. It's done on a national scale. It was with the National Opinion Research Center and investigators at University of Chicago. They did face-to-face interviews with elderly individuals ages 57 to 85, and they had about 800 variables. There were a ton of interviews on relationships, sexual behavior, housing, medication, just overall mood, social life, activity. It was so much data on just anything and everything they asked. Um, I only looked at round one, the core data, and I stratified by sex and ethnicity. And so this is what I got. So this is just from the cross-sectional data or the original. I stratified by Hispanic men, Hispanic women, Black men, and Black women. And these are the feature ranks selected by Explana. It's colored from 1 to 10, light to dark. And then after that, it goes from light to dark and grayscale, just because the colors were getting confusing when you had too many colors there. So I wanted to really focus on the top 10 features. That way you can see where those are. And then over here, these are just colored by, I should have included the legend here, but it stands for a relationship category. So it's like felt lonely, difficulties piling up, felt lonely. That was the number one thing related to happiness. So happiness was the response variable I used. And here, taste ID, I still haven't figured what that one means. That is I think it's something to do with mental health and things, but when they have difficulties with identifying bitter and sweet, and there's something going on there. I need to look into that one more. But then there's basic needs, how well their building is kept. Is that important to their happiness? Has at least 50,000 in assets, basic needs, relationship. But one of the interesting things, just because it was so surprising, I thought, a lot of these people had, these groups, the stratified data, they had mental health, relationships, physical health. I kind of expected those. I didn't know how important sexual health would be to elderly individuals. But it was only sexual health came up, these categories, mostly for Hispanic men. And then I was talking to a friend about that. Here is a closer zoom in on that. And I wanted to point out the percent variation explained. So how well all the data explains the response variable was lower in this, and it will be low in messy data or data that just doesn't exactly explain your response. We're seeing that in a lot of the microbiome data. Some is very high, but a lot of it will be closer to 10, 20%, and you're just trying to rank the most important variables. So it might not do, you wouldn't want to use it to predict surgery if you're getting a really low percent variation. But you can use it to rank to a certain point to rank the most important features in your data set. So these were these were decent, but these were the most important variables. Again, like I said, sexual health comes up for Hispanic men. This is a little close up of the other ones. And this was an interesting feature. So I saw that the frequency of intercourse, how often they were having sex. The reason, not just to be spicy in this talk, the reason I'm bringing this up was because this is a clear example of how, so on the left, you have more happy would be these predictions right here. This is another type of shot plot. And then less happy is over here, down at the bottom. And these categories, this is how often they have sex. Not applicable, don't know, refused, never, barely, sometimes. Few put sometimes, no one said rarely or never, none of the Hispanic men. Most of them, or not most, a lot of them were saying always. and a lot were saying not applicable. And you can see this reverse trend with how often they're drinking. So per week. So people who were saying that they were always having a high frequency of sexual intercourse and drinking a lot had a higher impact on happiness. But when they had said that it was not applicable, this right here, this is at the bottom, that had the lowest. So it was having the biggest negative impact. So people who are not having sex and drinking a lot might be the most unhappy. That's just kind of, what's coming out in this, how this might help you make hypotheses, and just how these plots help you see how the data is impacting the response or the variables. I thought that was pretty interesting, this trend. And then this comes out with masculinity. There's, I was talking to a friend and she was saying, there is a lot of pressure on Hispanic men to prove their masculinity through sexuality. And there's a lot of cultural, they were saying specifically penetrative sex meant that they were more masculine. So there could be pressure that might make them feel very happy. And then this was an AARP survey, because I couldn't find a ton of information and I don't know how to do this research. Like I said, this is not my expertise, but this was saying that Hispanics, this AARP survey they did, they found that Hispanics placed a higher value on sexual intimacy in their relationships, and they're more likely to agree that sex is critical to good relationships. So that was interesting. And when I looked at Hispanic women, I saw this pattern with Epstein-Barr virus, where higher levels of EBV were related to a negative impact on happiness. And that was really interesting. This clear pattern here, EBV is related to, so higher levels of EBV are related to lower happiness and less EBV. And so this is something, it's one of the most common viruses worldwide. People get it often in childhood, and most people will get it in their life. And the CDC said most people will not have symptoms, but is, depression, it is a symptom. So, and this is in elderly people. So I thought this was especially interesting that they're still showing levels of virus, and it's related very clearly to happiness. So, and EBV is associated mononucleosis with a risk factor for depression. There's all kinds of data on this, mood disorders potentially. And then there is a vaccine coming in. And then this is another data set I looked at just off of Kaggle. There's a lot of data, which I'm excited to kind of explore and play around with Explana. This was just a divorce data set. They looked at 150 couples in Turkey. They used the Gottman. It's a famous, I don't know what you'd call that, but this is a famous quiz test. They can predict, the Gottman Institute, they predict whether couples will remain together or get divorced pretty accurately. Like, results have been shown 90-something% accurate. They asked them 50 people, 55 questions, and they said never to always on these questions from 0 to 4. And I did actually get 94% from, this is from the Explainer report, using just 300 trees and Bermuda-Shap, to find statistically significant variables. 170 people were included, and tells you how many were accepted. A lot of them, these variables were important. So, a lot of the variables were accepted. And this is less likely to divorce, likely to stay together. This is just, so then I looked at the top two questions, just out of curiosity. The top two questions that were the most important were, my spouse and I have similar ideas about how our marriage should be. And the second one was, my spouse and I have similar ideas about roles in marriage. So I just thought it was interesting to point out that the most important variables were not, I have a great time with them. It's just maybe, but I'm not able to make for sure hypotheses about this topic, but it was interesting. And again, back to some code stuff. While I have some time, I'll save some time for questions. But again, I mentioned you just need this little configuration file. That's what a user would see. But on the back end, I'm using SnakeMake, and it's a workflow manager. And what that did was help me be able to use all R and Python, because my lab was, and a lot of people so I noticed that at the maybe it was where I was at, Anschutz Medical Campus or the area, everyone used R. I barely heard anyone talk about Python. And so maybe out of pure pressure, or I just started to use R a lot more. And it is very powerful. I actually really like it for a lot of reasons. I really struggle with graphics and Matplotlib. I have to figure that out. Um, but so what the SnakeMake allows you to do is make these rules, and it handles a bunch of different scripts for the workflow. So each one, this is pre-processing, dimensionality reduction, integration, making your delta data sets, and each one allows you to use a different script. And these kind of parameters come from the configuration file. So it's taking this data, sending it to your snake file. So that's the main file that you use. And it's passing all this information around from rule to rule to rule. And it's figuring out what it can build, and it can parallelize things really easily. So if you're doing different things at different times, or if you start at different places, I didn't write it too much like that, but you can do a lot of fancy things with SnakeMake. This is the random forest. So this is where the figures, those parameters where you set those things. That's in Python. And so this is right here, you can see this reference. So this is where this is actually what would go there is the original, first, previous, or pairwise. And so it knows that it needs to build analyses or do analyses for each one of those models. So it builds them all. And then at the end, it does some processing, and then it builds a report. So that's kind of where you're getting right here. First, original, first, previous, and pairwise. And each one of these folders, so this is each time Explain-It is run, you get a whole folder full of data. But I didn't want to just pile out data. I wanted to make a report. So that's why I made that interactive report. So I kind of went over how Explain-It streamlines feature selection. I show the delta data sets can uncover important features depending on different contexts of change, including the order dependent categorical features. It had good performance on simulated data, kind of breezed through that, but that's in the paper. It's designed for microbiome analysis, but it has really broad application because if you use it as a feature selection tool, it can be used on a lot of types of data with continuous numerical outcomes or categorical, binary categorical for the most part. It can be used with categorical. So if you had text values, it can still be used. I made sure that will work. Or you could code it as 0 or 1 if you had more than 2 classes. Sorry, you could code it as 0 to ongoing if you had multiple classes, and it would just be continuous basically. So future directions, get the paper published. It is in review at Bioinformatics right now. It's been there a month, so I'm hoping, we'll see. If it doesn't get accepted, I have some feeling on why it wouldn't, but hopefully it will get accepted. I have a website, because right here you can see, and you guys can sign up. I haven't even sent one email. I'm not going to spam you. But what I have right here is a link to, so it's free for academic use, and you can license it now. So if anyone knows of any commercial applications or companies that would like to use it, they can license it through my university. But this analytics services just links people over to my website. So I built an LLC, and it's set up through Wix with booking and everything. People can just go book an appointment with me, and I can analyze their data. And it's set up because I wanted a way for people so academics can use it for free, but a lot of labs don't have a bioinformatician. So I wanted to just have a way that they could just contact me and be able to get their data analyzed. So that's why I did build the LLC, so that I can work independently. And that's so, but this right here, figure out how to maintain and develop the tool. It's a bulky tool. And since it has so much broad application. It's not like the tool I developed in Scott's lab where it was only designed for fungal diversity analysis. It was so niche that not everyone is going to use it, but this tool has such broad application. I need to know how to maintain it and develop it after it gets published because I can't do it all alone. It could be improved. Yeah. So yeah, if you guys have any questions, let me know. Let's see.
That was great,
Thank
Jennifer.
you.
Really fascinating how
Thanks.
how you came to decide to do your project in some ways because of all the struggles you had wrangling data. I mean, and then you came up with this, what looks like a magnificent solution to that problem that actually, other people can use, can even start a company. I mean, that's really cool. So I'm happy to see it. So I'm going to start with a couple of questions from the students, and one of them is actually similar to what I had. Arun is asking, what percentage of success of a longitudinal study were all participants, what, like, where, how many of these longitudinal studies are participants there all the way to the end? And I think another way to say is, what's with your missing data? Like, how does your program deal with missing data? How does that affect your features in the end? And do you actually have that as a, like, oh, I can tell you that missing data is a huge problem in this one feature, or how does missing data work?
Yeah, so missing data. So so if it's a categorical variable, you would have just not applicable in there, and it would just become its own category. So one of the things I added is just hard to mention all these things. It does what's called one-hot encoding or binary encoding. So it'll take categorical data. And so if you had a category like the pill color, right? So it starts off as pill color, blue or green. It actually turns that into multiple columns, is blue and is green, yes or no, 0 or 1. So it'll convert it to a numerical value. So then if you had an NA column, it would just convert it to a different category. So it can handle a lot of the missing data that way. You do have to impute. That's still kind of something I've been looking into more. You can do, simple imputation like averages for that column if you're missing data. Or you can do more complex methods like using mice or there's some other ones I wanted to look into more. But then when you don't have an individual for that final time point, you would just not have a data point for that, right? So then it just becomes a question of how you handle your imputation. So you could either look at smaller time points, like if you were missing a bunch of people from the last time point, but you think your study was really interesting most of the time, you could just not look at that. So the nice thing about having the scripts written in you can script in the configuration file is that you could just say, okay, don't look at the last time point because I'm missing too much data. So it's very flexible. Like you can look at different chunks of time if you need, or different groups. You can stratify it really easily, like I showed with the different gender and ethnicities. So yeah, they can
Yeah,
handle.
I'm writing a paper on imputation, and one
Oh,
of the things we noticed is, of course, like and we had a bunch of different imputation methods like KNN, random forest. There's like dozens of them. And one
right.
thing we noticed that we showed that was, as you get a higher level of missingness, the worse your imputations are. So, I don't know.
Yeah.
There's not much you could do about it, but it's something that worth exploring and figuring out. So let's go to another question. This is more about your company. So Jin Shem asks, can you tell us more about your company? Are you a co-founder? Are you part of any incubator? Have you thought about those things?
No. So Explana is not a company. I started Jennifer Fouquier LLC to just allow myself to do analytics for anyone who wants help, because I didn't want to just keep this at one company necessarily. It's so broad that I just wanted to be able to help other people analyze their data. I am interested in doing things with Explana, like starting. So the website is really just a website for the software. I'm going to eventually add demos to the website so that people can see all the cool things it does. But it's just a software website. There's no explain a company yet. I'm interested in that, but that's a whole, I guess I get an MBA at. No, I'm just kidding. I don't know. But I'm interested in that, but I didn't know how to go that route yet. And I am worried that if I start with, I have a few conferences that I'm going to, and I'm going to try to talk to some microbiome companies, but I'm a little bit worried if I work with just a microbiome company, is that the best path for the tool? And so I don't know yet. That might be the way to go. Start with microbiome because that's what I'm
I
familiar
think
with.
like clinical should be the way to go. I mean, clinical people have a lot of money too. You know?
Yeah, and if it's used for hypothesis generation, then that's great, and they could look into some of these things if they want to do it for that. But yeah, I mean,
That's a good
so I'm open to any suggestions. If anyone has any ideas, please email me for sure. I'm open to ideas there. Yeah.
Um, I had another question, sort of similar. Well, Northern, what was your inspiration to change your field from industry, and why did you come to SDSU?
Oh, well, so I've always wanted to stay in I like industry. But I wanted to get the skills. I wanted to learn about bioinformatics. I wanted something that was that made me more just specialized because what I was doing in the lab, the last thing I was doing was working with microarrays, and I would get all the gene expression data and then I would give it to my boss, and I didn't get to look at all the data. So I was just like, all right, this is not fun. So I that's why I went into bioinformatics. But I actually really like industry because I like having a team and I like the pace. It's very fast. But I have always liked both sides. So that's kind of, I think this tool is fun because it could be used in academia and industry. And that's why I have it set up free for academic use if they want to help. It, that's what it's designed for. And then I want to protect it. That's why I went through the tech transfer process at CU Innovation so that it's protected. It's not free for commercial use.
Yeah, Arun has to run, but I'm going to
Yeah.
continue to answer some questions. Thanks, Arun.
Yeah.
Yeah, so just a question that someone had in the chat was I think it was for your graphics library. What did you use? Let's see. What was it? Oh, curious what library you used for the visualizations in the interactive report because the plots look so cool.
Oh yeah. So the plots are a lot of the plots are from other people, like Shap. Those Shap plots were developed by his first name's Scott, I believe, Lundberg. So that those are very helpful. I mean, they have all kinds of different plots too, and it just helps you see how your variables interact and impact the response. It's for interpretable machine learning, and that SHAP can be used with all kinds of different machine learning methods, not just random forests. So those are really amazing. And the Baruda SHAP plots that tell you how the variables are, that comes from Baruda. Those are just bar plots, box plots. And then so I use R Markdown, which is how I was piecing together all of the plots. So I just put all of the PDF images from SHAP. I did that in Matplotlib, which I'm horrible at, actually. That's been a struggle. But putting all the PDFs into one file, that way when people open up the report, they have all of the images in one set spot in one long file. That way they can scroll through and look at it rather than opening up, click, 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 having 50 different plots open. And so the R Markdown really helps you get this interactive, clickable interface. So you can add tabs. If you look at R Markdown, they go over how you can have different tabs, different sections. You can have links for the directories. That's what the Explainer report really has.
That's really
Different
cool. Yeah,
sections to browse through your data. I
yeah,
should have gone over a little more of that, but
we use R a lot. I talk about Python. Of course, my students learn Python, and it's an amazing language. It's better, I think, for if you want to really make software and integrate stuff. But we use it all the time for data analysis, and it's very powerful. But I loved about SnakeMake, and everyone should know this. Like, you can do any kind of script that you can run.
oh, right.
You can run it on the command line. You can run it in R. It could be R, it could be a C program, it could be, and my students put together these SnakeMake pipelines that are really really
Yeah.
cool and very, very easy to put together because you see all the inputs and all the outputs and exactly where the files are, and it's really a good way to go.
Yeah, I wish that I would have learned it a long time ago and just had a basic snake make workflow for myself so I could run different things I was
Hear that,
running.
everybody? This is why we got this recorded video. You can always go look back and look at those
Good.
images to see what a snake make workflow can actually look like. If you don't mind us looking at your code.
No,
You
and I mean,
know,
this is all open source.
yeah,
The code is open source. So you guys, it was a big project and there was a lot of things going around. So
right.
I wouldn't say it's the cleanest code. There, I want to go back. Scott would be so mad to see how little I documented my code, which I do regret actually. That is a big regret because I was going so fast and I was working with all kinds of different problems and I didn't document my code as well as it should be. And that's hurting me. Not
Yeah, no, this is
it
great.
is
Like
hurting
everyone
me.
listen to that. We should amplify that
It's
because
true.
it's you think you're documenting it for everyone else, but actually sometimes you're really documenting it for yourself because you look at it 2 years later and you're like, why the heck did I do this? And if you
Yes.
don't have that written there, you don't know why you did that.
Yes. And there are sections of my code where I look at it and I say, this is the weirdest thing. This has to be wrong. But then I go, but I knew that I knew more at that point. I wouldn't have done something that weird. So there must have been a reason why. So I wish I would have written more because I did some strange things to get this to work. And it should have been documented better.
How
So
long
I
have you been
definitely.
developing Explain It? That's what Jasmine was asking.
Oh, I was so I it's been a little while. I mean, it started like 2.5 years. But it there was a lot that went into it as far as just brainstorming and thinking and kind of
Sure.
just it took I just kept running into all this data. And what I was also having a problem with is just collaborating and communicating. Someone would say, well, I told you to filter that, or I don't want to include the immune cell populations here, or and I would just have to keep organizing my data and I would get so confused. Like, I don't know how to explain all this or talk to people. But now it's just in one report that people could share and collaborate with people and say, this is what it is. It's here, it's explained. The configuration file that I showed a couple times, that gets copied immediately and put into the folder. So it's saved for you. And so it's even in one of the tabs. So if you need to look and you say, oh, did I filter out that thing that it's saved there so you can see what happened. And then I can't believe I forgot to mention this, but it creates a dynamic methods section of the report. So it tells you written what you did so that you can copy and put that into the methods section so that it just says we used 300
Great.
random 300 trees and all your parameters and it includes that.
Oh yeah, that's great for publications
um
too.
yeah, so it was designed with academics in mind for sure.
Yeah, and reproducibility. I mean, and I think
Yeah.
that's an industry thing that you picked up is
Oh
like
yeah.
notes came back in my lab. You're just like out notebooks were key. Everything was in a notebook. Everything was reproducible. Like you just you came coming from industry was really helpful. Okay, I had
Yeah.
for you about yeah. It's more of a comment about cause and effect because
It's
features are. It's
poor.
really interesting to look at the features, and in most of your cases, you're just like
It's
you know,
correlation.
yeah, exactly. And it's really clear with the COVID stuff because you're like, well, COVID is actually the cause of people's illness,
Right.
But it's interesting because you're like, well, but it's like all these things they're not an effect of COVID. They're are which one is responsible for you dying? It's sort of an interesting hypothesis. Like if you could, for instance, if you could change one of them, oxygenation would be a classic where you could actually save someone's life by giving them more oxygen because ultimately that might be the problem, right? Or that's obviously you can't have not have oxygen. But it is really interesting to think of cause and effect when you look at features and explain it doesn't let you know that. And it's sometimes very clear that it's not a cause and effect. It's really an effect, not a cause. But sometimes it's not so clear.
Yeah. Yeah. um yeah.
It's just a comment. I don't know.
Yeah,
It's just like
no.
it's just really interesting to say like, for instance, someone might be really depressed and they might have this problem, but it could be that they're not, say, having sex or whatever because their job sucks. right? Because they have
Right. Oh yeah. Oh right.
every night, and the real source of their depression isn't that, but it's actually because they can't find any time to like have a relationship with anyone, or they can't see their
Oh
partner,
yeah.
or whatever. So anyway, it's something everyone should keep in mind about these things. It's like, you get these features, and then you have to go back and test, if you can, whether that's the act. If you alleviate that one feature, whatever it is, does that change the situation? So
Right.
anyway, just a comment. But if you don't know what the features are, you can't do that. And that's why Explain It is so cool.
Yeah.
So yeah, we're up to 107. I think we should
Oh,
probably call. There was some more questions. People are very interested in your work. I would say that I'd be happy to
email.
talk to you anytime about the compositional stuff. Um,
Yeah.
I
Sure.
think I'm glad that worked out for you. And I do remember, I mean, I know why theoretically it should work, like why it should help, because it makes the data less dependent. But the fact that it sometimes doesn't is really interesting, and it would be really cool to look at, that, and maybe even imputations, because we're starting this conversation. But to look at, why exactly, when does it work better? Like under what conditions
Right.
is it, and how does it help? That would be a very cool study.
I wrote it in an email so that I would have it documented because it was weird when I saw, I can't remember exactly why, but I saw at one point it made it worse.
Yeah, and why is that?
And
Under what conditions, and why would that be the case?
and that
Why?
was with simulated data.
Yeah, yeah, with
That was when I
simulated
was
data.
looking
How
at
does that?
simulated data, and so I was really confused and I said, but it was something in the parameters that I was changing. So um
Eh,
I have to look into it
fine.
more.
Yeah, and I definitely think is plan. Like I love the fact that you point out the MRFs, which is really cool, and the SHAP stuff. We haven't used that, so I'm really
Yeah,
happy.
it's really helpful, but it's complicated with all of those different tools. That's why I made the workflow. It's just too many
Yeah, we have to use them all
things.
independently, and you put them all together nicely. So, and that is great. Well, we'll have to get together again soon, Jennifer,
Yeah, we will. We
to
should.
talk about all this and bring you to
Thanks for
life.
having me.
Yeah, no, I can't.
If anyone has any questions, feel free to email me.
Yes, it's right there at the bottom, Jennifer, at explainit.io. And thanks so much again for meeting with us. And this video will be online. So I will,
Thanks.
if you ever want it to, you let me know.
Oh, perfect. Thank you.
Bye bye.
Bye.